Good morning friends, I am Shomajit sir. Today I am here to deliver my lecture on topic DNA fingerprinting. This is from the chapter Molecular Basis of Inheritance. So watch the entire lecture to get basics out of it. So without any delay, let's start with the topic DNA fingerprinting. So let's start with the topic DNA fingerprinting, also called DNA profiling. So you can see DNA fingerprinting or DNA profiling. So it is a technique to identify the similarities and differences of the DNA fragments of two individuals. If two individuals are there, we can differentiate by the DNA fingerprinting method. And it is developed by Alex Jeffrey in 1985. So you can see the picture in the right hand side of Alex Jeffrey. Next we'll see the basics of DNA fingerprinting. First is DNA carries some non-coding repetitive sequence. A number of repeats is specific from person to person. So number of repeats in a gene is specific to a person to person. So person A, you can see this genes have 8 repeats. Person 2 is also there. Person 3 is also there. So person 3, you see it has a 9 repeats. Okay. So you can see the repeats. First one is 8 repeats, second one is 9 repeats, and third one is 10 repeats. So we know that there are many repeats in DNA, so it varies from person to person and it's specific. So you can see the nucleotide here, the gene here. You can see the bases. First person has 8 repeats, next one 9, and third one 10 repeat. Now, repetitive DNA can be separated from bulk genomic DNA as different peaks during density gradient centrifugation. So, the bulk DNA forms a major peak and the small peak are called satellite DNA. Okay, so the bulk DNA forms a major peak and the small peak are called satellite DNA. So, you can see a uniform one having a density gradient centrifugation, and when the uniform one is centrifuged, it's called, you can see, gradient one. Now you can see microsatellite, that is also called short tandem repeats, that is STR, and another one is there mini satellite, that is VNTR, variable number of tandem repeats. So satellite DNA is classified as micro repeats, not micro repeats, microsatellites. Okay, so satellite DNA is classified as microsatellites, mini satellites, etc., based on the base composition. If A and T reach, okay, if adenine and thymine are rich, or GC is rich, and guanine or cytosine is rich. Is the length of segment and the number of repeat units. A DNA sequence which is tandemly repeated. Okay, so a DNA sequence which is tandemly repeated in many copy number is called variable number of tandem repeats. So what is VNTR? DNA sequence which is tandemly repeated in many copy number, that's VNTR, and VNTR belongs to a mini satellite DNA. So in mini satellite, you can see the size of each repeat is hundreds of base pair. Okay, and in micro satellite, the size of each repeat is 2 to 6 base pair. Next, you see in a person, the copy number varies in each person. Two alleles, that is parental and maternal. Okay, so two alleles are there. One is paternal and maternal of a chromosome, also contains different copy number of VNTR. Okay, so two alleles, that is paternal and maternal of a chromosome, also contains different copy number of VNTR. So you can see the copy number of VNTR, parental one and maternal one. The paternal and maternal one, two alleles of the chromosome. So you have to remember here, microsatellites have 2 to 6 base pair repeats, and many satellites have hundreds of base pair repeats. Next we have VNTR is specific from person to person, and the size of VNTR varies from 0.1 to 20 kb, that is kilobytes. Okay, so VNTR is specific to person, and the size of VNTR varies from 0.1 to 20 kb kilobytes. So any differences, okay, so any differences in the nucleotide sequence, inheritable mutation observed in a population is called DNA polymorphism, variation at genetic level. So what is DNA polymorphism? It's a variation at the genetic level. So polymorphism is higher in non-coding DNA sequence because mutation in this sequence may not affect the individual reproductive ability. So these mutations accumulate generation to generation, causing polymorphism. So polymorphism have a great role in evolution and species. So you can see DNA polymorphism, that is single nucleotide, that is SNPS. So these are the steps of DNA fingerprinting. The first, we'll see the steps of Southern blotting technique. First is the isolation of DNA. That means first we'll isolate the DNA, that is from any cells or blood or stain or semen, stain, saliva, root hair, bone, okay, or hair roots, skin. So these are the different samples from where we can isolate the DNA. One is blood, one is cells, one is stain, one is semen, stain, saliva, hair roots, bone, skin. Okay, so you can see from these things we can isolate the DNA. Next is the digestion of DNA, that is how we can digest by restriction endonuclease. So by restriction endonuclease will fragments each of the DNA. You can see you can fragment the DNA segment. You can see DNA fragments with sticky end. So restriction endonuclease can be sticky end or blunt end. So the most used one is the sticky end, where they produce a sticky end notch. Okay. The third is the separation of DNA fragments. How we can separate by gel electrophoresis. So then treat the DNA fragments with alkali solution, that is NaOH, to denature DNA bonds in the gel into single strand. So you can see the gel electrophoresis. The fourth step is the transferring or blotting of DNA fragments to synthesis. Okay, so transferring or blotting of DNA fragments to synthetic membranes such as nitrocellulose or nylon. You can see the nitrocellulose membrane, as well as you can see the electrophoresis. The southern transfer, see, you can see the paper towels are there, nitrocellulose are there, gel membrane or the sponge are there, tray with buffer are there. So this is a process of southern blotting. And next step is step 5, that is hybridization using radioactive level VNTR probe. Okay, so you can see here, place a nitrocellulose filter paper in a solution of radioactive labeled single stranded DNA probe. Next, DNA probe binds with the complementary sequence of DNA fragments on the membrane to form a hybridized DNA. Okay, so you can see the radioactive VNTR probe, which will wash and it will form VNTR probe hybridized to DNA fragments. Okay, so first is we'll take the nitrocellulose paper, and then we'll make a radioactive VNTR probe, and then we'll wash it. So you can see the DNA is being hybridized. So in the sixth step, you have detection of hybridized DNA by autoradiography. That is, hybridized DNA is photographed on an X-ray film. So you can see the hybridized DNA on the left hand side. Then we have X-ray film. Okay, so the autoradiograph gives an image in the form of a dark and light band. It is called DNA fingerprints. So you can see the dark and light bands in the X-ray film. So DNA fingerprint differs in everyone except the monozygotic, that's identical twins. So DNA fingerprints differ in everyone. The sensitivity of the technique can be increased by use of PCR, that is polymerase chain reaction. Therefore, DNA from a single cell is enough for DNA fingerprinting. Because we have PCR, if we have a small sample of DNA in by means of PCR, that is polymerase chain reaction, we can amplify the copies of DNA. Okay, So DNA samples are their primers, their nucleotides are their tag polymers, are their mixed proper, are their PCR tubes, are their PCR cycle. So you can see the PCR cycle, first is denaturing, then handling, and then extension. So this is one PCR cycle, numerous PCR cycles from. So you can see the temperature in the denaturing area, it is given 95 degrees Celsius, and the annealing part is given 55 degrees Celsius, and the extension part is given 72 degrees Celsius. So in case of a PCR, one DNA strand is there, double stranded. It is being denatured at a 95 degrees Celsius. So DNA strand will be separated, and then next the annealing part, the primers will be added in both the DNA templates, and then there will be extension at 72 degrees Celsius. New DNA synthesis will take place. So in a PCR, from its small DNA segments, we can produce amplified amount of copies. So this is the DNA fingerprinting at a glance. You can see first is the isolation of DNA, second is the digestion of DNA by restriction in nucleus, third is the separation of DNA fragment by gel electrophoresis, fourth is the transferring DNA fragments to synthetic membrane, fifth is hybridization using radioactive labeled VNTR probe, and sixth is detection of hybridized DNA by autoradiography. So you can see the pictures on the left hand side. The first one you can see isolation of DNA, second one you can see the digestion by restriction enzyme, the DNA is broken down, third is gel electrophoresis, you can see the long DNA fragments and short DNA fragments, fourth is a transfer it to the membrane, and fifth is the hybridization with the label probe, and sixth is the detection using X-ray. So what are the applications of DNA fingerprinting? First, it is used in forensic tool to solve paternity, rape case, murders, etc. It is also used for diagnosis of genetic diseases. Third is to determine phylogenetic status of animals, and fourth is to determine population and genetic diversity. So these are the very important application of DNA fingerprinting. First, it is used in forensic tools. Next, for genetic disease treatment. Third, for phylogenetic status, and fourth is to determine the population and genetic diversities. So you can see here one example is given. You can see a solving paternity using DNA fingerprinting. Here a person 1 is likely to be the father of the child. Okay, person 1 is likely to father of the child. You can see first one is the mother strand, second one is the child chromosome. Okay, the third one, which one will be the father? You can see father 1, 2, 3. So you can see the first one is matching. First father 1 DNA fragments is matching with the child. So we can tell that your person 1 is likely to be the father of the child. So you can see here solving a crime with the help of DNA fingerprinting. So you can see there chromosome 7, chromosome 2, chromosome 16. These are there. These are paternal, maternal one. So DNA from individual A and DNA from individual B. So you can see the chromosome 2 is there, 7 is there, and 16 is there. So DNA from the crime scene we have obtained chromosome 7, 2, and 16. So you can see amplified repeats separated by the size on a gel gives a DNA fingerprint. Okay, so you can see the matches. Okay, the C is matching with the B one. Okay, so thank you, students. Thank you, students. This was all about the entire video lecture. Here I've discussed about the topic DNA fingerprinting. This was from the chapter Molecular Basis of Inheritance. Thank you. Again, we'll meet in the next class. Thank you, students.